So I'll just introduce you to everyone. So everyone who's joined in, thank you so much for joining in and welcome to the Own Your Future Mentorship in July. And we've had a break over the last 2 months. So we're happy to see everyone join in. Thank you so much for attending. So today we have 2 amazing speakers, Dr. Dalila Burin, who's a cognitive neuroscientist, and she's specialized in bodily consciousness and sensory-motor integration in healthy people and neurological patients. She did her PhD in neuroscience at the University of Turin in Italy before joining NIRx as a scientific consultant in 2024. She worked in Japan and Ireland as a professor and researcher, combining her expertise in virtual reality with brain imaging techniques, including fNIRS. So thank you so much, Dalila, and she will be the first speaker for today. And then the second speaker is Dr. Elina Zmeykina. Sorry if I pronounced that wrong. Elina holds a PhD in theoretical and computational neuroscience from the University of Göttingen, Germany. Her doctoral research focused on applying TMS, EEG methods to investigate neurological conditions such as disorders of consciousness, stroke, and depression. And she subsequently completed a 2-year postdoc at the Department of Cognitive Neuroscience in Göttingen, exploring heart-brain interaction and how cardiac signals influence the consciousness perception of TMS-induced phosphenes. Currently, she works as a scientific consultant at NIRx Medical Technologies, where she supports and advises researchers in initiating and advancing their work with fNIRS technology. So thank you so much. We're really delighted to have 2 amazing guests on our show today, and we really value your presence today. So over to you, Dalila.
Thank you very much, Juhi, for the lovely introduction. I'm going to start by sharing my screen. There you go. Well, once again, thanks very much for having me, having us. I'm sure I can speak also for Elina when I say that we're very happy to be here. It's 7 p.m. in Berlin. Nonetheless, we're very happy to be invited here and to join this series of very, very fascinating events. Today, we will focus on fNIRS, functional near-infrared spectroscopy, and TMS applications, in particular in the context of translational clinical research. Before starting, allow me to very briefly introduce us as a company, NIRx Medical Technologies. We are based in Berlin, but we have a network of global distributors, basically all over the world. We have been around now for 25 years, and we specialize in providing high-end fNIRS systems and solutions to researchers such as yourself. We do not only manufacture the hardware component, but also, of course, the software. And we also pride ourselves in the amount of support that we actually provide, not only to our customers, but also to the general audience, and in terms of like educational events, seminars, webinars, just like essentially we are doing here today. I will start with the, let's say, first part of the presentation, which is the focus will be mostly on fNIRS, and then Elina will take over. She's our in-house expert regarding TMS applications. So I'm going to start from the very beginning, the very own definition of fNIRS, functional near-infrared spectroscopy. F, what does it stand for? It stands for functional, which emphasizes the fact that we are looking at functional activation rather than the structural one, although, of course, also the structural component is quite important. Near stands for near-infrared, which is the specific type of light that we use in fNIRS, and we are going to see later in detail which kind of specific light. Lastly, S stands for spectroscopy, which is the study, the method of sending light into a substance and reading it out on the other side. So in summary, functional near-infrared spectroscopy, fNIRS for friends, is a form of non-invasive functional optical imaging that measures the hemodynamic changes at the level of the brain cortex, of the surface of the brain. When a person is performing a certain task, whatever that is, it can be a cognitive, a motor, a social task, whatever, there is an increased neural activity in the specific portion of the cortex that is directly related to the execution of that task. Consequently, there is an increased demand for energy, for metabolism. Neurons don't have their own reserve of energy, so they have to take it from the bloodstream in the form of oxygen. So the system responds through the so-called neurovascular coupling by vastly increasing the local supply of fresh blood flow. So essentially, once the blood flow reaches the necessary regions of the brain, the hemoglobin, which is a protein that sits in the red cells that essentially carries the oxygen, releases the oxygen, and therefore, once oxygenated, continues its path back to the lungs. This specific increase in fresh blood flow and volume to specific areas of increased neural activity forms the temporal and spatial specificity of the hemodynamic response, which is exactly what we measure in fNIRS. Just to give you an idea, this graph on the right is essentially how a perfect, let's say a typical pattern of a hemodynamic response looks like. We see that first of all, we have 3 lines. This is because the hemoglobin, the chromophores that we measure are actually 2. Oxygenated hemoglobin, usually displayed in red, and deoxygenated hemoglobin, usually displayed in blue. Combined together, they give us the total hemodynamic response, the green line. Something else very important that I would like to highlight, perhaps some of you in the audience may have already noticed, is that the timeline, so basically the x-axis in the graph, you see that is displayed in seconds. This is because the hemodynamic response is by nature relatively slow, certainly slower compared to, for example, an EEG recording, where I'm sure people are more used to milliseconds time frames. Here we're in the order of seconds. This is because from the onset of the stimulus, again, it can be anything, but from the onset of the stimulus, for the hemodynamic response to peak, it takes a few seconds, generally between 4 and 7 seconds, sometimes a little bit longer, to peak, and then the same amount of time to get back to the original baseline. This is something particularly important to consider, especially when we design an experiment with fNIRS. I mentioned before very briefly that spectroscopy is the study of the interaction between light and matter. When an incoherent source of light, it can be any kind of light, even the light that is illuminating our rooms right now, when an incoherent source of light, white light essentially, is sent through a prism, the light is split into the different wavelengths, or better, the different visible colors for us. Let's say, for example, that we are interested in one specific wavelength, a green wavelength. This one wavelength is sent into a tissue or a material, and it's read on the other side. The difference between the light that's sent in and the light that is received on the other side can determine important information about the structure that's in between. In fNIRS, in the specific case of fNIRS, the tissue is the human brain, and the light that we're sending in is near-infrared light. The changes in light absorption reflect the hemodynamic activity, the hemodynamic response. To better understand this process, we need to see in particular how the photons, which are the light particles, propagate. When, again, there is a source of light, which is, again, practically any kind of source of light, but also the sources of light in the fNIRS system, photons travel in all of the directions. Basically, they shoot off of the light source. But when they have to pass through a tissue, which is a diffuse medium, light is partially scattered and partially absorbed. Absorption happens when photons hit a molecule, is destroyed, transfers its energy, And so basically it doesn't exist anymore. It ceases to exist. It's no longer passing through the tissue. On the other side, scattering, it refers to when a photon either reflect, or diffract off of and through the tissue. This is actually when the detectors come in play, catching the photons that are not absorbed on the other side of the path. Now, one of the problems is that we don't know completely and exactly the full path, let's say, that these photons take, but we can somehow predict it and control it. In particular, for the photons that reach a detector, we can determine their average path, and this path is going to take the shape of these, you see this yellow 3D shape probing the tissue between the source and the detector. This shape, yellow shape, we actually called photon banana. I did not make it up, it's a real scientific term. And it's not a coincidence that it's yellow, but this is how it's called, the photon banana, because of this peculiar shape, let's say. And it's essentially this 3D shape that probes the tissue between and underneath the source and the detector, which is precisely what we need in our fNIRS measurement. Another problem actually is that for every billion of photons that we send in, only one reaches the detector in the adult brain in a, let's say, generic configuration. Which means that in order to receive a significant amount of photons, we need to send in billions and billions of photons, and also to have detectors that are sensitive enough to detect them. But what are the specific wavelengths to detect hemoglobin specifically for fNIRS? Here you see in this picture, there is an incoherent source of light, like a white light, sending light through the human tissue. In one case, it's a hand. In the other case, it's a human skull. And we see that the color that we see from our side, basically, it's red, which means that red long wavelengths, presumably, can actually go through, they can penetrate the biological tissues. In particular, these red lights include wavelengths from the visible, but also from the infrared range. So it's kind of just in between the edge of the visible light, and that's why we call it near-infrared light. So presumably, in our fNIRS system, we need to use these red long wavelengths because they are the ones capable of passing through the biological tissues, but which ones specifically? To answer this question, we need to look at how molecules in the tissues absorb the different wavelengths of light. In this plot on the right side of the slide, you see that basically on the x-axis we have all of the possible wavelengths, while on the y-axis, we have the so-called absorption coefficient, which is essentially a measure of how good each molecule is at absorbing light. Now we see that the wavelengths between 700 and 900 nanometers seems to be the ideal target for oxygenated and deoxygenated hemoglobin only, and nothing else. If we go a little bit further over 900 nanometers, we see that water starts to significantly absorb light, and the biological tissues are mostly made out of water and lipids. If we look at the absorption coefficient for, again, oxygenated and deoxygenated hemoglobin, we see that there is a peak of absorption at 760 nanometers for deoxygenated hemoglobin, and a peak of absorption at 850 nanometers for oxygenated hemoglobin. With these two wavelengths, specifically 760 and 850 nanometers, we can safely and distinctly target deoxygenated and oxygenated hemoglobin only. And that's why our system, just like the vast majority of the fNIRS systems available in commerce, they use these two specific wavelengths of light. I briefly mentioned before that in spectroscopy, when we study light, we usually have a source of light that, of course, emits light, and a detector that receives it. In the specific case of fNIRS, sources and detectors are called optodes. The pair composed by a source and a detector is a channel, which is an essential component, an essential unit in the fNIRS system. Channels detect the light that is scattered in between and underneath the distance between source and detector. To be exact, the depth of the photon banana, you remember the shape, the 3D shape probing, basically the surface of the brain, in between the source and detector, the depth of the photon banana essentially depends on the spacing between the source and the detector. Spacing optodes, sources and detectors further apart leads to a greater depth of image. But of course, if the spacing is too big, not enough photons will actually reach the detector. And that means that your signal is going to mostly be dominated by noise, essentially. By contrast, by spacing sources and detectors very close to each other, we are going to have, of course, a higher signal to noise ratio, but also a more shallow depth of the image. Generally speaking, let's say the general rule is that the depth of the photon banana is half of the distance between source and detector. In a general configuration, it's a standard configuration, in particular in the adult brain, the optodes are always placed at 3 centimeters of distance, which means that the depth of the image will be around 1.5 centimeters, which is essentially just enough to probe the cortex, essentially, of the brain. This is a standard, let's say, general configuration, but of course, the exact distance and the depth of your channels in the fNIRS system depends, of course, in what you're interested in your research, of course. Also on the hardware, because not all of the fNIRS systems available in commerce are exactly the same. But generally speaking, there are 2 levels to consider. One is the topographical one, and the other is the tomographical level. From a topographical perspective, essentially placing sources and detectors at equal distance from each other. So again, 3-4 centimeters in the adult brain, a little bit less in children, around 2.5, of course, because of the different head size, but also because of the different thickness of the skull. In this case, essentially, we don't discriminate too much for the depth of the image because all of the optodes are basically at the same, almost at the same distance. This is definitely the most popular form of imaging when we talk about fNIRS. It's relatively easy. It's quite easy also to process and to analyze. But of course, there are limitations. And the biggest one is that you don't consider for the depth, of course, of the image. And that's where tomography comes in. This is essentially when you place sources and detectors at different distances from each other. So it can measure essentially different cortical depths. Now, this is actually not a very common procedure, let's say, in fNIRS, with a couple of important exceptions. One of them is short channels. Now, when I said before that in the photon banana, basically the light, the photons, travel from the source of light, then they probe the surface of the brain, and then eventually they are detected by the detectors. To be precise, actually, before reaching the surface of the brain, they actually have to go through several layers before reaching the actual brain. They go through the skull, they have to go through the skin, they go through several layers and back, of course, to be detected again by the detectors at the level of the scalp. So the problem is that our measurement includes, of course, the, let's say, actual hemodynamic response, the brain-related hemodynamic response, but also signals coming from outside of the brain. Also, of course, at the level of the skin, of the skull, there is a certain level of physiological activity, mostly due to the fact that we are dealing with human alive people, like heart rate, blood pressure, or respiration. And of course, this spontaneous systemic physiological activity can contaminate the fNIRS measurement, and it can significantly impact our data, especially when these systemic changes, physiological changes occur together with the cortical activation. So for example, when there is a lot of physical activity. The idea with the short distance detector, with the short channels, is to specifically measure and therefore then to regress out these extracerebral component. In our system, in particular, we place sources and detectors very, very, very close to each other. According to the literature, the ideal distance is 8 millimeters in the adult brain and around 2 millimeters for infants, which is practically impossible to achieve. But essentially, short distance channels can capture exclusively and selectively the extracerebral hemodynamic activity. Is not really hemodynamic activity, it's just physiology. And therefore, we can, in the post-processing phase, regress it out from the main, let's say, the entire measurement, and we can end up only and selectively with the real, let's say, task-related hemodynamic activity at the level of the cortex. Just to give you an idea of how important these the measurements coming from the short distance channels are, here you see in this graph, I hope you recognize the kind of typical pattern of the hemodynamic response. So this kind of like curve that peaks a few seconds after the onset of the stimulus and then goes back to its original baseline. You also may have recognized that the oxygenated hemoglobin and the deoxygenated hemoglobin, they kind of go in this counterintuitive pattern. But you see that there is also some kind of fluctuations or something that is not exactly perfect, let's say, as the first picture we saw. This is because this measurement here still includes the short distance measurements. And this is the very same picture we regressed out the measurements coming from the short distance detectors. And you see the picture we have is a very clean, almost perfect pattern for the hemodynamic response. So far, I have been speaking practically exclusively of light intensity, but how do we get from light intensity to hemoglobin concentration? With a continuous wave fNIRS device, which is essentially how the vast majority of the fNIRS available in commerce work. So essentially by emitting 2 wavelengths of infrared light continuously at a constant intensity. As I briefly mentioned before, the problem is that photons are scattering around and we cannot completely account for the entire path that they take from source to detector. We cannot completely account for the entire scattering process. So what we do to bypass, let's say, this problem is that we don't measure absolute values, but rather concentration changes with respect to an empirical baseline. Generally, this baseline it can be a period away from the main measurement or an average of the entire period. But the important thing to understand here is that we don't measure absolute values, but concentration changes in the hemoglobin absorption of the light. So I was saying, basically, to go from the light intensity to the hemoglobin concentration, there are a couple of steps we have to go through, and they are mostly just formulas. I know they may look scary, but they are actually literally just a formula. And in most of the softwares available to analyze fNIRS data, including the one that we provide from our company, it's literally just one click away. The first step is to determine the optical density, which is essentially this formula you see here. So basically the negative logarithm of the raw light intensity over the mean, the average of the raw light intensity. So it's a kind of like a normalized measurement of the light intensity to determine the optical density. The second step, to actually get to the hemoglobin concentration, we need to apply another formula. This is called the Beer-Lambert law, or better, the modified Beer-Lambert law. Again, it looks scary, but it's actually not that scary. And it's literally just one click away. Now, for educational purposes, I'm going to very briefly explain it, but just so you understand the process. We have the optical density, then the formula also includes the extinction coefficient, which is kind of like what we said before, how good each molecule is at absorbing light at specific wavelengths, so lambda. And basically, the formula has to be repeated twice for the two wavelengths. D in the formula is the distance between source and detector at the level of the surface. We said before, in a standard, typical, let's say, configuration in the adult brain is 3 centimeters, 4 centimeters. And lastly, the last factor, important factor in the Beer-Lambert law is the differential path length factor, which is essentially a multiplication factor that accounts for the fact that the photons don't go straight from source to detector, but they actually take the famous photon banana path, which essentially means that they travel 6 or 7 times more than the actual distance from source to detector. And in fact, typically the differential path length factor is 6 or 7. So essentially we are ready now to, we have seen basically how the fNIRS works, how the hemodynamic response looks like, how the light is sending, goes through basically the surface of the brain, and it's detected on the other side. And the difference essentially in terms of light intensity gives us information on how much hemoglobin there is, either oxygenated and deoxygenated, in a specific portion of the brain while the participants are performing a certain task. fNIRS is just one of the many neuroimaging modalities. I'm sure here in the audience there are people familiar with other systems, with other neuroimaging systems. So how does fNIRS compare with other imaging systems? In terms of spatial resolution, the gold standard, let's say, is probably fMRI with a resolution of about 3 millimeters. And of course, it can scan the entire head, not just the surface, of course, of the brain. Although we know that fMRI has also a few disadvantages, a few limitations, let's say. The temporal resolution actually is not particularly high. It's generally in the order of 1 to 3 hertz. While in fNIRS can go up to 10, 20, sometimes even 50 hertz. It depends on the configuration. fMRI, we know it's extremely expensive, not only to purchase, but also to maintain. While other modalities such as fNIRS, EEG are generally more affordable, of course. And probably the most important and significant difference, fMRI doesn't allow for practically any kind of movement. It's necessary to stay completely still inside the scanner, which means that also certain populations cannot perform tasks, experiments in the fMRI, while systems as fNIRS or EEG, they can be portable, and they are generally very suitable for pretty much any population, going from children to neurological and clinical populations, or young adults, or pretty much anyone. It's also possible, of course, to record from different modalities at the same time. So, for example, EEG and fNIRS at the same time. This is because fNIRS is very compatible with different other solutions. For example, EEG, TMS. Elina will speak about that later in the presentation. But also fMRI too, with, of course, the appropriate hardware, the appropriate setup. This is particularly convenient, of course, because fNIRS allows, well, for movement, first of all, is very suitable, as I said, for pretty much any population, and still is extremely robust in terms of recording and data quality. participants' comfort. As I said, it's also very suitable for very small babies or elderly and so on, which makes it ideal for very ecological, naturalistic, and child-friendly, for example, research. What can we do with fNIRS? With the limitations that I mentioned before that are in the nature, let's say, of the hemodynamic response, pretty much anything you can imagine and something more. In particular, for the purposes of today's presentation, I'm going to mention just very briefly a couple of studies that are I find particularly interesting and very fascinating. I hope you will too. I know that the audience today is quite different, so I selected a couple of studies, recent studies that I think might be curious and interesting for the purposes of today's presentation. But in general, there are a lot of clinical applications, of course, for fNIRS. As I said, going from neurodevelopmental populations, even in children with developmental disorders such as ADHD, autism, but also for clinical applications such as motor rehabilitation or neuro rehabilitation in neurological conditions, but also psychiatric situations. In this one study, in particular, that I selected, that it has been published like very, very recently, just a few weeks ago, we see, well, I think a compelling application of fNIRS because they record from intensive care unit patients, in particular patients with acute disorder of consciousness, hospitalized in the ICU, to assess volitional brain activity, covered volitional brain activity. Here, researchers aim to identify signs of consciousness in patients who appear unresponsive using a relatively simple motor task, or better, motor imagery task. Participants in particular were instructed to stick out their tongue, or at least to imagine or to try to do that, because as I said, they were mostly behaviorally irresponsive. And so the researchers here, they used fNIRS to detect any kind of activity, volitional activity, residual activity in the prefrontal cortex, which we know is, generally speaking, an area involved in the voluntary planning of actions. What is fascinating, I think, and remarkable is that in a subset of these ICU patients, they were actually able to identify significant hemodynamic response during the mental imagery task, despite no behavioral evidence of awareness, which suggests that there are still somehow preserved cognitive functions, even in these patients with acute consciousness disorders, and even in absence of an actual motor output, which also means that fNIRS is a very suitable system for delicate patients, even bedridden patients, such as in this case, but it's also sensitive enough to detect these minimal levels of, in this case, consciousness, or better, motor imagery related to the consciousness. They used in particular, actually, in this study, our portable device, what we call the NIRS Port 2. But in this situation, of course, there was no movement. It was actually a very static situation because the patients were completely bedridden, which is also a little bit challenging sometimes because of the mechanical placing of the fNIRS system on the head. So perhaps I thought it was nice to show you also this second study that investigates how physical movements impact the executive functions in children with ADHD. In this case, if I remember correctly, quite a large sample of children with ADHD performed the Stroop test, which is a very common test for executive functions, the one where you have to say, for example, the word red written blue, you have to say blue as quickly as possible, not red. In this case, the children were supposed to perform this task, the Stroop task, in two main conditions, while sitting still or while moving. And at the same time, the prefrontal cortex activity was recorded. We know that actually children with, in general, but also in particular with ADHD, very often move. They cannot really sit still completely, in particular during the execution of certain cognitive tasks. So researchers actually found that movement can increase the prefrontal activity in these children, and even more in particular in those with the from the inattentive subtype. Something else that I found quite curious is that this effect was even more pronounced in children, in boys in particular, and those with higher ADHD symptom severity. Once again, this is just a very, let's say, simple, relatively simple example of how fNIRS can be a very effective brain imaging method, even in delicate populations such as ADHD children, with overcoming some of the most typical challenges, for example, the movement limitations that you have, for example, in an EEG setup or an MRI setup. And instead, actually, it allows completely for movement and allows the exploration for the cognitive effects of the presence of movement or not in a very delicate population. Again, in this case, actually, they used our portable device, the NIRS Port 2, the one you see in the picture here, which is particularly suitable for situations where there is a certain level of motion. With that, I'm sorry, I'm slightly over time. I conclude my first part, let's say, regarding the introduction of the fNIRS. And there are a few resources here that might be, I hope, useful for you for eventually get to know more about fNIRS. Just so you know, you don't have to remember everything. We're gonna probably share the slides that we presented with the organizers, of course, of these webinars. So you're gonna have access to basically all of the slides and the information that we gave you today. With that, I stop sharing my screen and I give the control of the webinar to Elina.
Thank you, Dalila. Let me share my screen now. All right. Hello, everyone. And I was already introduced before, and I'm Elina, and I'm very happy to present today my favorite probably research topic is TMS fNIRS. As you heard, I have some background, some research background with my PhD and postdoc with TMS, and TMS fNIRS is something that I'm very excited about. So let me start probably from some basics of what is TMS. So TMS is Transcranial Magnetic Stimulation, and that is a non-invasive brain stimulation technique which allows us to activate some neurons in the cortex just by applying the coil over the head. So it's a non-invasive method, it's painless, and it's usually very comfortable for participants. So it's used to investigate and modulate neural functions of different neurological or psychiatric patients and also of healthy population. At the moment, transcranial magnetic stimulation and its different protocols approved for major depressive disorder as a treatment alternative to some medication or some medication-resistant patients, but it's also not approved but widely used in the clinics for different stroke rehabilitation, for multiple sclerosis, schizophrenia, addiction, and all type of neurological diseases. And TMS is a technique. Here on the picture you can see how usually the treatment or experiment looks like. The patient or subject is sitting in a chair, and we use the coil to produce this strong magnetic field. And the TMS, of course, has so-called limitations in a sense of the maximum stimulation depth. So usually it's limited up to 5 or 6 centimeters in depth. And we are activating the neurons. So coil has some wires inside, and we produce a very strong magnetic field. This magnetic field in its turn can induce some electrical current in the underlying cortical tissues, so-called neurons. So that's how we actually activate the neurons. And then the pulses of TMS, they're quite short, and we can there are a lot of different parameters that we can vary, and it means that the application of TMS is very, very wide. So we can basically fine-tune it based on our needs of brain activity. And usually I would like to split two type of TMS application based on experimental design. And first, I would call online TMS effect, and that means that we apply the TMS intervention or some pulses of TMS right after some baseline assessment, and we measure the effect of the TMS pulse immediately after. So it means that milliseconds or seconds right after the pulse. We can repeat it several times and then evaluate the effects of online TMS. And then the second group of studies, also based on some experimental design, is an offline TMS effects. And in these cases, after the baseline assessment, we apply the TMS intervention, and it's not necessary just one pulse or some train of the pulse. It can be a protocol which lasts for, 20 minutes, 30 minutes. And then we do the assessment, or we evaluate the effect not immediately after, but after the application. And in this in this group, the timing doesn't really matter that much. So not to the precision of milliseconds. We can evaluate this effects minutes, weeks, or even months, and that is the type of experiments that we usually use in the clinics. So here the most important is whether the TMS had any clinical effect and clinical improvement or not. And TMS, the studies that I mentioned, they can be used with TMS device only. So if you just have TMS machine, you can run any stimulation to any region of the brain and estimate the effects. What kind of effects do we have? So usually we measure the cortical excitability, and in this case we use some EMG which we put on the hand, and we measure the so-called motor evoked potential. So this is due to corticospinal excitability. When we activate the motor cortex, we are able to induce some twitches in the head, in the hand. Then another TMS alone effect is the TMS-induced phosphines. This happens when we stimulate the occipital area, and when we are on the right spot where we can induce those phosphines directly on the visual hotspot, then we can induce those very short and very bright flashes of light. Then also one of the most common clinical TMS application is the functional mapping. So this is usually used, for example, pre-surgical evaluation, when you can use TMS and apply the pulses around the region which one assume can be tumor, and you can see before the cutting out this part of the brain, you can check whether the areas will be affected, the functional areas will be affected. And that is very common in the speech areas mapping. Then there are also some intracortical interactions which can be also measured with MEP with both hemispheres, and some behavioral or clinical assessment, which is usually the clinical scales or clinical evaluation of the treatment. TMS has proven its effectiveness, so it's applied in many different clinical areas and also research areas. However, what is known about TMS that it's high variability. So even small changes in frequency, intensity, or duration, they can lead to different outcomes. So it doesn't really mean that TMS effects are unpredictable. They have some trends, but TMS has quite high variability. And also even some identical parameters and responses, they can vary across individuals and within one individual between the sessions. And that is because when we apply only the stimulation, only TMS, we make an assumption that brain is a static black box. So we don't really care and we don't know what is happening inside the brain. We just apply our pulses and hope for results, hope in a sense in the clinical improvement. However, of course, brain is not a static system. It's a dynamic system. So there are a lot of processes happening in the background, and different parameters like timing or brain region or some anatomy or some general excitability level can lead us to different results or different outcomes. And what is actually happening in the brain, TMS alone cannot answer. So we can induce some activity, but what is happening there, we can tell exactly, and we can only assume. And I have already mentioned some TMS parameters which we can tune, and here I want just to cover some basic or the main parameters that usually people take into account when they do the TMS research or the TMS clinical application. The first is a target. So this is quite straightforward. So we want to stimulate functionally relevant area. If we stimulate or study the motor cortexes, we are searching for motor area. If we want to do some cognitive tasks based on the research, we would go for prefrontal area, so dorsolateral prefrontal cortex. And this is quite standard. However, the TMS, what exactly TMS activates? So there is some kind of a pattern in the brain. So it depends, first of all, on the coil shape. So we have different manufacturers have different coil shapes, and it results in the different shapes or different profile of electric field distribution in the brain. And the latest are usually the 8-figure shape coils, and they're much more precise and they're much more focal. So this is became much more popular now in the TMS application. The maximum stimulation depth is 1.5 to 2 centimeters inside the cortex, and people also use the neuronavigation to control where exactly we stimulate. So as it's very precise method, we are able to control even if we apply the stimulus or if we activate the neurons on the crown or on the wall of the gyros, or which exact gyros we do activate. And neuronavigation system can improve results of TMS very well. Then another TMS parameter is an intensity. So intensity is usually related to the percentage of the individual's resting or active motor threshold, or its minimum intensity to evoke MEP. So as I already mentioned before, the MEP is the metric to measure the motor evoked potential, and this is at the moment is a gold standard to set the stimulation intensity. So, for example, in the clinical application of rTMS for depression, there is a protocol where you choose the stimulation target and stimulation intensity, and intensity is based usually on the percentage of motor threshold. So first you measure the motor threshold, then you set the stimulation intensity, and you move the coil not to motor cortex but to the frontal area where TMS is applied. There are other methods like based on phosphines or some fixed stimulation percentage, and also one of the latest is induced electric field. So some researchers use some computational models to take into account some of the anatomy of the brain, also coil shapes and some technical parameters of TMS devices. Motor work potential based stimulation intensity has some pro and cons, and of course it's an immediate feedback. It's very easy to measure. You just need two electrodes and find the hotspot. It also reflects the excitation and inhibition of the motor cortex, and it became so-called the gold standard for TMS. However, it has some cons, and first of it is that it's highly variable between participants, but also between the sessions and days. So everything like, for example, if a participant or patient was biking to the lab of experiment, or how much coffee they drink, if they smoke, if they drank alcohol the day before, everything can vary excitability. And those kind of factors, those are just several factors that are known, but also there are many of them which we don't even know. And it can change the cortical excitability and therefore change our methodology. So the thing that we are based on, so MEPs is a method to set the intensity. And also there are a lot of technical challenges. It's sometimes it can be a little bit challenging to find the hotspot or to find the exact amount of stimulation intensity, or it can require too many trials. But also one of the main is that it lacks physiological justification for non-motor areas. So in this way, we don't really take into account anything like the cortical excitability, the cortical organization, and some anatomy, and anything like this. And we just move this intensity to different brain areas and assume that it should have the same effect as it was in motor cortex. And two last parameters are the frequency and duration, or they are all together form so-called protocols. So as the pulses are very short, we can apply them with a great variation. So it can be either a single pulse or repetitive TMS with high or low frequency, or even some bursts with 20 milliseconds in between. And those are protocols that are usually used in the clinics, and they are believed to be excitatory or inhibitory based on their after effects. So to summarize the first part, TMS is a great method, and it can effectively activate some brain regions. However, the outcome might be variable, and because we cannot really say what is an ongoing brain activity inside the brain. And also the methodology of TMS still lacks some physiological justification when we define the stimulation targets or intensities. And then how can we address the gap? So usually latest application of TMS is together or combined with neuroimaging. Neuroimaging is the observational approaches such as EEG, fMRI, or fNIRS, which can help to tell what is going on in the brain right before the stimulation, during stimulation, or after. But then when I checked some research on concurrent application of TMS together with EEG, fNIRS, and fMRI, which are the most common, it's clearly that EEG has a leading place in this research because of the form factor. However, I checked, of course, some earliest publications about the TMS and fNIRS, and it was quite challenging just due to the technical limitations of the fNIRS optodes. People had to adjust and adapt all the coils or some stimulation locations or optodes. Everything has to be modified. And TMS fMRI is still a niche applications of TMS together with neuroimaging, just also because of the technical limitations. It is quite challenging and requires a lot of advanced techniques and hardware adjustment to be able to stimulate inside the scanner. And then TMS fNIRS. fNIRS has very little amount of publications just because of those early attempts. However, now with the modern type of the optodes or devices, application of TMS fNIRS quite close to TMS EEG in terms of the form factor. And TMS EEG and TMS fMRI together with so EEG and fMRI together with EEG, with TMS, they have a lot of insight, and they can bring a lot of insight about the brain activity ongoing or right after the stimulation. However, of course, they have trade-offs. So EEG is quite sensitive to electromagnetic noise, and fMRI is just expensive, loud, and static, and it doesn't really allow us to stimulate any brain region. And fNIRS stands somewhere in between. It captures the real-time hemodynamic, quite similarly to fMRI, and changes are linked to the neural activity. And it's also ideal for longitudinal and clinical application. There are some other TMS fNIRS advantages, except of the practical setup. So here on the picture you can see how the setup basically looks like. There are like flat optical fibers, and you can place the TMS coil on top of them and stimulate and record everything at the same time. So there are no special requirements for TMS coil needed, so it can be integrated with all TMS devices. Another advantage is that TMS and fNIRS, they work on the same depth. So fNIRS is region specific, and it's not confounded by whole brain volume effect. And on the other hand, TMS is quite spatially precise, so it doesn't really activate the whole brain, and both TMS and fNIRS, they work at the same spatial resolution. Also, fNIRS has advantages for temporal resolution to capture not only the hemodynamic responses but also physiology. And on another hand, hemodynamic response is quite slow, and it's not really affected by the muscle twitches, which we usually see on EEG. And one of the main advantages of TMS and fNIRS is, of course, the fNIRS immunity to electromagnetic artifacts. And then I would be happy to give you some overview about what kind of mechanisms there are in TMS fNIRS, but so far there is not really enough research, and we can only assume that fNIRS captures some neurovascular coupling, similarly to any other fNIRS application, but also fNIRS can capture the local metabolic demand. So both in fMRI and fNIRS studies, we can see some initial rise in deoxygenated hemoglobin, which can be due to local metabolic demand and some dilation of the vessels. And fNIRS, also because it's very flexible and it can cover and record the signal from the whole head, it can also capture some network activation, some broader hemodynamic changes. And if you are interested in current state of TMS and fNIRS, I want to just give you this 3 systematic review about the current state, and I will show you just a couple of papers to show how people usually use TMS and fNIRS together. So one of the study in consecutive TMS fNIRS application for depression patients, the RTMS protocol was applied for 20 sessions, and that's a standard TMS protocol. It's 10 Hz RTMS stimulation over the left dorsolateral prefrontal cortex. And in between of sessions, the researchers, they measured the fNIRS during some cognitive tasks, and what was important in this paper as an outcome, that TMS could predict that for some patients this protocol did not really work. So already after the 10 sessions we could see that some people who are responders to clinical treatment, they showed the higher HBO activation comparing to those who did not respond to the treatment, and they didn't respond also after 20 sessions. So that can be a very valuable tool just to predict the outcome already in the middle of the treatment. Another series of the study from Thompson was the aim of the study was just to see whether we can calibrate the TMS coil placement, the angle and the intensity based on the hemodynamic response. So they did 3 studies where they measured the different coil placement, like angle of TMS, then the amount of pulses, but also the intensity. And they could distinguish the optimal TMS coil placement based on the hemodynamic response. So for example, the 45 degrees, the one that is used usually for clinic, it also resulted in higher hemodynamic changes. The same for amount of pulses and also for intensity. And one of also important study of concurrent TMS and fNIRS is where they used 2 types of defining the TMS target, and they used standard MEP guided method, so where they were searching for hot spot of motor walk potential, and then the second one was fNIRS guided hotspot. So they basically ran the study with some imaging of finger tapping, and they saw the highest activation, and they used it as the hotspot for the treatment. And what is important here is fNIRS navigation had some better outcomes for some clinical assessment, so some clinical scales were improved based on the fNIRS targets. But also what is important is that fNIRS guided hotspot was obtained also for patients for whom MEP guided hotspot was unpredictable or undetectable. Because there are some groups of patients like with stroke or with some damages in corticospinal path where we cannot really detect the motor walk potential, and fNIRS method was very useful here. And then one of the latest studies from 2025 is the study of one of our customer from Hong Kong, and they did ITBS, so intermittent theta burst stimulation over the left dorsolateral prefrontal cortex, and they measured the fNIRS signal under the coil, and they saw quite interesting pattern in increasing the hemodynamic response of deoxygenated blood and decreasing oxygenated blood right under the coil. So the results are quite interesting. It is still, their studies continue, and they will find out what was the reason of this interesting activation. And the study that I mentioned, the last one, they used the Borealis, and this is the laser-based system, fNIRS system, which you can use where the electronics on the head is not permitted, usually like it's used in fMRI and also in TMS. And for the optodes, slightly different from what Dalila presented you before. We have an optical fibers where we don't have any electronics on the head and all the electronics is outside. And that's how it looks like closer to the on the cap and the setup overall. So they are flat, they are around 10 millimeters, 10, 11 millimeters height, and coil can be placed just on top of them. And so just to summarize, TMS fNIRS is very practical. So flat optodes, they allow for seamless TMS fNIRS application. So we have no artifacts, no electromagnetic interference, and no need for specialized TMS hardware. fNIRS is highly relevant for clinical application. fNIRS can explain and also predict the treatment efficacy, offering a biomarker for clinical outcomes. fNIRS can also bring some neurophysiological insights. It can capture the hemodynamics right before the stimulation to inform TMS, during stimulation, right after, immediately after, but also to measure some long-lasting effect. And fNIRS can also bridge some TMS fMRI application or translate some TMS fMRI to a portable, cost-effective usage. And the last but not least, TMS fNIRS is still a new research field, but it's a growing field with high potential for novel discoveries and clinical application. And with that, I would like to finish, and I would like to encourage you to subscribe to our newsletters. We don't really spam with too many emails. We sometimes announce some webinars or our workshops. We do a lot of practical workshops everywhere in the world, including the concurrent application of fNIRS together with TMS, EEG, and all type of multimodal application. And thank you very much. With that, I will switch to...
Thank you so much, both of you. I personally really enjoyed the talk, and we already have a few questions from the audience. So Sadia, I think I will unmute you. You can maybe raise your questions to Dalila and Elina. And Eva, if you have any questions or anything to say. Hello.
Okay, first of all, thanks so much for a wonderful session. My question was that how TMS and fNIRS are combined actually is hard to visualize for me because I just want to know how can we use them. But is it TMS used after or what is the procedure? It just doesn't, yeah, I just didn't get that part. And also I'm was confused about does TMS waves damage the normal wiring of brain? Yes, it's also the question.
I'm sorry, I didn't hear the last part of your question. If you can repeat
The
it.
last part, yes. The second question was that the TMS wirings are very dangerous for the normal wiring of the brain. So it might damage the normal wiring of the brain.
Okay, if that's dangerous. Okay. So the TMS and fNIRS, they practically very easily combine. So what you usually do is you measure the fNIRS signal from your scalp and you place the coil on top of it. So there are no really restrictions or any limitations of concurrent applications. The only one can be just some mechanical artifact. So when you produce some TMS pulse, you have some kind of a coil vibration. So the limitation there mostly just practical. So I don't say that it's just very simple to apply. Of course, some training is needed, but without it can be applied without any problem so far. And about the damage of the TMS. So TMS is considered as a safe method for brain stimulation. However, of course, it has some limitation and some side effects or risk factors. And usually when like for research, for example, when we include some participants, we always have a list of exclusion criteria, and they're usually like the pregnancy, the history of epilepsy, or history of epilepsy of this person or in family, and in general some like constant migraines and something like this. So normally it's safe if you follow those recommendations for the exclusion criteria.
Thank you,
Okay,
Sadia.
thank you so
Does
much for answering.
that answer your question completely? Yeah.
Yes, thank you so much.
Okay, thank you so much. Right, is there any questions from anyone else? Okay, I don't think there's any questions from anyone else, but I just wanted to say thank you so much. You've given us so much knowledge about fNIRS, both the scientific explanation and also some clinical applications. And thank you so much for both for such an informative sessions. And Dalila, especially appreciate the discussion on the ICU applications. And I wanted to, I noted that you said that it is fNIRS is portable, and the patients in ICU, they're pretty much static, so it's like kind of the perfect use case for this type of technology. Previously, when I worked in pediatric ICU, it was such a heavy mission to get patients from the ICU to MRI. So something that I was wondering is, could fNIRS possibly be used to detect or monitor cerebral ischemia? So I understand that the limitations could be probably the depth, but do you see any use for this?
Yes, definitely. I think we have several researchers all around the world working on, well, stroke, but also ischemia, but in general, neurovascular disorders. And I would say in particular, not that much in terms of diagnosis, also a little bit in terms of diagnosis, but mostly in terms of recovery, let's say rehabilitation. So assessing, for example, certain functions, especially, of course, the ones relatively easily that you can relatively easily record from the cortex. So let's say motor functions or big cognitive functions, and how essentially they change over time, improve over time, ideally, of course, following a neurovascular event. So, for example, a stroke or ischemia or something. This is definitely something that's it's been done for a while now, considering fNIRS is a relatively recent technique. But it's certainly a very good application for that because of the feasibility, as we mentioned before, of the setup. It doesn't have to be necessarily used in a clinical setup. It can be easily transported, just like you said, which is a big advantage compared to, for example, MRI, where you have to bring the patient to the machine and not the other way around. Also, again, of course, there is the limitation of the cortex, but if the target is generally like, let's say, major cognitive functions such as motion, especially of the upper limbs, or I don't know, attention or executive functions, or visual or acoustic perception, then it's definitely something that's been used for a while now, also in a clinical context of neurovascular disorders. Yes.
Thank you so much. I can't wait to see it being used and distributed more widely, because I really think that this is quite an invaluable tool. And yeah, I've learned so much, and also about TMS. It's I really love that it's non-pharmacological. That's really, amazing in terms of mental health, especially. So yeah, I've learned a lot today. Eva, do you have any questions or comments?
Yeah, I mean, I also really learned a lot. Like I'm always working on my MRI topics, and then I'm always very fascinated, like about these techniques. Yeah, okay, that's my very simple question, but can it be like combined with MRI? I know people do like EEG stuff and MRI. I mean, I'm not working on these things, but yeah, maybe that's a short answer.
It is, yes. Now it's also quite a new area, like fNIRS, fMRI, and it's actually quite promising because they complement each other very well. fNIRS has quite high temporal resolution, and it's able to capture all the physiology which fMRI sometimes doesn't capture. So, for example, on fNIRS signal, we can see the heartbeat quite clearly, right? Which we take out later on. But the thing is that fMRI data, they also can have it. But just due to the temporal resolution or sampling frequency, we can't really clearly see it and can retract it. So fNIRS can be used some sort of like short channels, or can be like a regressor for fMRI data. And there are already some applications like this, when fNIRS data were used to feed in the into the model of fMRI, and the results were much more improved, much more cleaner, like without all this fNIRS physiology. So the application is quite
Yeah, that's really
interesting.
cool.
Yeah.
Yeah, that's amazing. That's really cool because it's really hard to combine these things sometimes. And also, I was wondering, I mean, of course, like also just thinking of fMRI, I mean, sometimes people apply this also to different regions. I don't know, I've seen also stuff in the spine. Like, is this all applicable in principle to like any region, or does this require special calibration or something?
I think it is so. It does require different calibration in terms of the source brightness. So for brain, like for head, why we have all those techniques like dual tips, the spring tops, and all these accessories is because of the hairs. So one of the main challenge for fNIRS is the hair, because if they're on the way, they just absorb a lot of light. And if we have, for example, from light to darker hair, it can be much more and more challenging for fNIRS to detect very good signal. And then on the outside of the head, when we don't have really hairs, we need just slightly different source brightness. We don't have those problems or like these obstacles like with hairs, and we don't need that powerful sources and detectors. But yeah, it has to be adjusted. But in principle, yes, it can be applied.
Oh, yeah, that's interesting.
But
Yeah, I was really wondering also about this, like how you calibrated that per person to also in terms of, I don't know, is there a lot of like reflection? I can see there's probably absorption, as you also explained, but then also reflection or something that might be different, or something per person. But I guess it's all like automated in the process, or not?
It is like there are different systems, of course. In our system, it's all done on the software level. So when you put on the cap, of course, you need a different amount of light for the frontal areas, for example, for the lobe where you don't have hairs, like no one has usually, and then on the other areas like occipital. So for good signal, you need different amount of light and different detector range. So what software is doing is basically it tries to optimize the source brightness so that the detectors are not saturated and are able to capture the signal at the same time. So it's done like when you put on the cap, the first step before you start the data recording, similarly to this EEG impedance check, we do the signal optimization to make sure that all the sources they are on the right level.
Yeah, that makes sense. Yeah, I've seen now, I was working with a group, they tried to do like fMRI of the eye, like of the optical nerve. I can imagine that is also an interesting area because it's very hard, basically. Probably it's not possible, but it was like, because it's moving and stuff. So as you just said, like temporal resolution and things becomes like very difficult. It's very hard to also keep the eye like still. So yeah, it's interesting. Yeah, I'm really curious, like where all this could go with your methods. Yeah, thanks a lot. It was really
Thank you.
very, very informative and very interesting. Thank you.
All right. Thank you so much, everyone, for joining in. I've just posted the link to the Own Your Future Mentorship Seminar Series playlist in the chat. And if everyone could kindly just fill in the seminar evaluation so that we know how best to serve your needs in the future. So the next talk will be in September with Dr. Samuel Gatau, and he is a nuclear medicine specialist. So he will be talking everything about nuclear medicine. So please join in and please share. The adverts will be going on to be disseminated in the first weeks of September. And yes, we'll see you soon. And thanks so much to our amazing guests. We really appreciate it. And yeah, as we'll chat more about how to disseminate these slides over email. Thanks, Dalila. Thanks, Elina. We really appreciate your presence today. And everyone from the audience also says thank you from Isaac and as well as David. Yeah.
It was lovely meeting you all. Thanks very much.
Thank you very much.
Thank you very much.